This is episode 280, the Stem Cell Podcast, Liver Regeneration with Dr. Meritxell Huch. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how to improve and for suggestions on guests. Today, we have Dr. Meritxell Huch. She's at the Max Planck Institute of Molecular Cell Biology and Genetics. She's on the podcast today to talk about her research on tissue maintenance and repair in the liver and the pancreas. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, this week we'd like to introduce Hepatic Cell News, provided by Stem Cell Science News. Hepatic Cell News summarizes all of the latest research, news, jobs, and events in the hepatic research field and delivers it right to your inbox every other Friday. Subscribe for free at www.hepaticcellnews.com.
All right, you guys, I'm kicking off the roundup today with my first of two stories that kind of speak to the plasticity that, I mean, it's a trend long coming. I'm not saying that we were still up there on the Waddington landscape, but I think this is an illustration, and these two stories that I have, as well as Arun's for that matter, are illustration of how our thoughts on cell plasticity are really evolving. This is a cell story with zebrafish. It's actually a resource that is kick-ass, and that's the best thing about this, is you guys could get online right now and go and do your own investigation, similar to the proof of principle that the authors here provided, showing how this utility, this resource, could be applied toward innovative concepts. So yeah, I mean, I'm alluding to it, but the challenge here is lineage tracing, right? And tracing individual cells as they organize at all, like the single cell, like micro scale as well as macro, in terms of like tissues, organs. And this has been a battle, a challenge that we've been facing for over a century now, since the beginning of light microscopy, tracking cells visually. I mean, that was kind of the first stage, and you could argue culminated or kind of hit its apex in terms of light microscopy in 1983. Where the complete cell lineage map of C. elegans was created just by observation, watching these cells move around. But of course, you know, there's a couple limitations there. One, you need to be looking at an organism that has very highly stereotypic cell fates, which is the case in the worm. And also like optically and like the density, the scale of the organism. So enter light sheet microscopy, which was a major sea change. Which enabled this in toto imaging of embryonic development. It's been applied, I would argue, most famously in the zebrafish, but also has been used in mouse embryogenesis, also Drosophila. And using that, you can look at strict temporal intervals and map and trace cell position over time using like fluorescent reporters, et cetera. Add to that, nowadays, you got the single cell seq, all the omics, which provides a really highly complementary approach of providing, you know, the transcriptomic and combining that with the spatial temporal to really give a map of embryonic development. But, you know, to date, consolidating this into like a one-stop thing, it's been elusive. This multimodal cartography of developmental lineages that incorporate this kind of spatiotemporal microscopy mediated, as well as transcriptomic landscape. You could say it hasn't been done to the full hilt. Enter the laboratory of Loic Royer. I hope I pronounced that right. Probably not. My apologies. At the Chan Zuckerberg Biohub in San Fran, and they did this. They combined light sheet microscopy with single cell RNA-seq, single zebrafish embryo-seq, as they call it. So they broke out embryos like one at a time in like a single cell set and made the libraries on those cells from embryos at successive stages. And all of this, I mean, the images in this, Arun, you've seen this paper, I'm sure. I mean, the images are bonkers, as you would expect from zebrafish. But also the incorporation of the transcriptomic data, right, is really critical, providing context and deeper, resolution of what's going on there. And it's all available for your enjoyment and perusal at zebrahub.org. So have a look at that. And how do you use it? And I'm like, you could go in there and create some insight for yourself, and then maybe extend that to your own system. I mean, zebrafish, I know they're fish, right? But they're vertebrates, and a lot of these processes are conserved. And here's a couple examples of how you can apply this to really advance the field. So these relatively newly identified progenitors called neuromesodermal progenitors. I mean, when I came up, the whole idea of neuromesodermal anything was kind of like, what are you talking about? These are distinct lineages. But of course, lo and behold, there are these neuromesodermal progenitors that yield skeletal muscles and posterior neural cells, marked by SOX2 for the neural end of that, as well as brachyury for the mesodermal end. And they've been identified in multiple systems, multiple organisms, including mammalian human cells. So here, looking at those cells and all the lineage trajectories combined with the spatial lineage tracing, they suggest, the data suggests that there was like a broader differentiation plasticity here, coming back to that theme of my papers, the broad plasticity of these neuromesodermal progenitors, and effectively that the early NMPs, as they're called, are like they're effectively pluripotent, which is nuts. And they show that by combining the transcriptomic data and positioning, et cetera, and all these kind of velocity and trajectories of differentiation. So that was one cool example. Next, they looked at these lateral plate mesoderm cells in early somatogenesis. And from those data, they challenged this classic lateral plate mesoderm lineage segregation model and showed that instead these cells were contributing to both the hemangioblasts, so these endothelial hematopoietic progenitors, as well as the developing pronephron, which will ultimately become the kidney. So these are, I wouldn't say like, I think people are receptive to this notion of cell plasticity, particularly early in development, and cells existing along a spectrum of epigenomic, restriction or permissiveness, whatever you want to call it. But still, so I wouldn't say this is like iconoclastic or like some kind of dogma breaking science, but yeah, this is counter to what we thought we knew. And it's all came out of a real close look at these data sets in this resource that you could go look at right now. And zebrafish is a great model organism, I think, fundamentally for all the the optical clarity and the accessibility, all that. But it's also, I think, a vertebrate, as I said earlier, where a lot of these processes of organogenesis, not all, are conserved right up to humans. So I think this is going to be an amazing resource that I'm personally going to look into and see if I can fish out something related to, gonad development that is really, really tightly defined transcriptomically, as well as with this lineage tracing spatiotemporally.
Fish out something related to organogenesis. Yeah, gonad development. I see what you did there.
Yeah.
Yeah, I see what you did there. Yeah, great resource for the community here in Cell. It's one of those Atlas stories, but like what we talked about before, the reason why these Atlas stories go to the next level is the application, right? They could have just done enough to be like, hey, we created this incredible repository, this incredible resource for the community, but like what you're talking about, they applied it for studying the mechanistic underpinnings of development in zebrafish. And I've talked about this a billion times. I love zebrafish. I've been a big fan of zebrafish ever since my days at the Duke University. Go Blue Devils. Had the chance to chat with Ken Poss early on during my undergrad days, and that was actually the big reason why I became a cardiac biologist, because zebrafish have this incredible regenerative capacity and an incredible regenerative ability in their hearts. kind of got me on the road to becoming a stem cell biologist. But I mean, those sort of observations in the heart and elsewhere are enabled by the fact that this is such an incredible model organism for development. We've talked about its exceptional properties that make it a great model system for development ad nauseum here on the show. But just to recap, of course, it's very amenable to imaging, as you're showing here. Beautiful, transparent body, especially early on during development, enables the intersection of new technologies like light sheet and all these really awesome confocal approaches that not just these folks are using, but also the POS lab is using at Duke. Um, yeah, it's got a fast generation time. It's easily manipulable genetically with morpholinos and all these other new approaches as well. So it's an incredible system. I think the only, in my mind, the other system that rivals it for its ability as a to be a great model system for development and genetics is the Drosophila. And funny enough, a lot of these imaging papers and network mapping papers have also been coming out in Drosophila recently as well. I don't know if you saw, there's a study looking at the connectome of the Drosophila brain and imaging that to like an incredible depth. And once again, that's just because fast generation time, easy to manipulate with genetics and so on. This is a really phenomenal resource. It's kind of a emblematic of what the Chan Zuckerberg Biohub can do. Very Silicon Valley kind of science, great, heavy data, heavy computational power required to develop something like this. But I think the stuff that's been coming out of the CZB over the years has been really quite exceptional. I don't know if I'm going to use this in my own work, but I'm just excited about the images, and I might just look at it for fun, just because I just love looking at zebrafish images. I've been doing it for the last 15 years.
Yeah, partner. I mean, I don't know the scientific value, but the eye candy is there. I'm going to take some of these pictures from my screensaver, 100%. And not even kidding. As you're talking about that connectome story from the fly, and I was reading this paper, I saw that one, and I see this, and like the deeper resolution we go on this, and like this kind of like programmatic element of development understood at high resolution, as you said, using these like this in silico science that is really heavily leveraged in these biohubs. Like, not even kidding, I could see it, how this is all a simulation, because like literally at the cell level, these cells, like you're figuring it out. It's like x plus y equals z, and more complex math than that. But you could see how all these events that we attribute to like the magic of Mother Nature are really just little cellular machines executing their program. So a little bit of a existential moment for me reading this paper. No fault to the authors, but you bugged me out.
My bad, but I didn't realize you're going to take it that way. We're living in a simulation, but hey, you're not the first person who said that to me in the last week. So who knows, man? Who knows? And we're using tools like these to get to the bottom of how stuff works in the simulation. Regardless of your existential crises, this is a great, great resource for the community. I think there are some limitations. One big one I thought was while it's a great data set, it's sort of limited and it doesn't capture the final differentiated state of the tissue. It doesn't go all the way to the end of organogenesis. Kind of, it goes from somatogenesis to the beginning of organogenesis. But I mean, hey, there's going to be other versions of this, I'm sure, coming down the pipeline. So stay tuned, and we're going to move on to something else that's in the realm of development. We're moving way, way earlier in development. We're talking about fertilization. And, we don't talk about this super early aspect of development a lot on this show. This is the Stem Cell Podcast. But what is, of course, the ultimate totipotent stem cell? That is the zygote. The zygote can turn into pretty much anything, right? So we're talking about pretty much how sperm and egg come together to make the zygote. And that's the process of fertilization. Speaking of cool computational tools, this story really heavily utilized AlphaFold, which is this incredible resource that's come out over the last few years to look at protein structure. And in fact, the founders of AlphaFold and some of the folks in charge got the Nobel Prize in chemistry, I think, for it this year. So it tells you kind of how applicable and useful this tool has become. I know there was some skepticism about AlphaFold over the last few years, last 5 years or so, but it's become really impressive in looking at protein structure, looking at protein conformation, binding, and all these different things. So that's one of the things that they did here. in the context of fertilization and figuring out the proteins that are responsible for the process. So we know about fertilization, of course, it's the basics for sexual reproduction, culminates in the binding of the fusion of the sperm and egg. And there are a lot of proteins that are, of course, involved and critical for this process in vertebrates. But funny enough, and I honestly didn't know this, I didn't realize that we hadn't figured out all the molecular mechanisms of how this works, and especially true across different species. So, understandably, different species you would expect would have quite different mechanisms when it came to the protein interactions during the process of fertilization. That actually it may not be so true, depending on the results of this paper. So here they use an AlphaFold multimer screen and identified a protein called TMEM81 as part of this conserved trimeric sperm complex with other fertilization factors called Izumo1 and SPACA6. So the really crux of this paper is this TMEM81 and how it's essential for fertility in zebrafish, like we were talking about, but also importantly in mice. So we're not just talking about fish, we're going all the way to quote higher vertebrates and the mammals, and showing that this TMEM81 is kind of conserved across that evolutionary tree of vertebrates. And in line with the trimer formation with this Zuma1 and SPACA6, they also show that these three proteins interact in zebrafish sperm, and that the human orthologs of those as well also interact in vitro. So again, conservation of these processes across millions of years of evolution. Pretty cool. And notably, this formation of this complex creates a binding site for egg fertilization factors like bouncer in zebrafish, and they looked at some other stuff in the mouse as well. This one called Juno in mammals. Kind of, they present this as like a lock and key kind of mechanism where these three trimer conserved proteins in the sperm bind to and basically unlock the egg in some way. But the egg part of it apparently is a little less conserved. So, like I said, there's that bouncer element to the fish on the side of the oocyte, and then Juno in mammals. But regardless, I think an incredible data driven and computation driven story here. The foundation was this AlphaFold algorithm, whatever you want to call it, that was figuring out how these three sperm proteins are actually interacting, and it computationally predicted how they would interact in the context of the sperm. And that was the foundation for the rest of the story. So pretty cool approach here. I think it's got a lot of elements. It's looking at conservation of different proteins in this process of fertilization across the mammalian evolutionary tree and the vertebrate evolutionary tree. Yeah, and just I think a lot of cool elements to unpack in this story.
Yeah, you know me, I love anything having to do with reproduction. It's right up my alley, and I love this story. A lot of really cool illustrations of that kind of lock and key mechanism, imaging wise too. So I thought this is definitely something you want to have a look at. But for me, it was kind of it felt like to me a use case study for the new world, this new era of in silico protein, modeling, prediction, and engineering. And I was so pleased that AlphaFold won the Nobel, the people behind it, because some might argue a little bit hasty, but I don't think you can overstate what a watershed this is. I can remember my very first class in graduate school. For a minute, I was at Cornell before I got severely depressed and transferred back to New York City. But in that first class of protein structure and function, Linda Nicholson, I'll never forget, she said, this is a problem that we're probably never going to crack. From protein structure from a primary sequence. And of course, I think she said probably. She didn't say never. And she wasn't wrong. But I don't think anyone could have anticipated how we've leveraged the computer technology. And I think it presages this new era that we haven't even seen the beginning of yet, of making proteins that don't exist anywhere, that are designed for specific functions, that can, marry and make complexes of things. I don't know. It's beyond our imagination, my imagination right now. But I think we got the beginning of this new era of protein engineering that this is kind of an insight into, and that you can go from this in silico to insight. I think is kind of the correlate of what we're going to see soon, and we've already seen some of just making a protein from literally from scratch, from design, based on principles. And I think that's going to be the next 20 years. Who knows? Generation time in science now is like 5 years, but I think for the rest of my life, I think that this is going to be a huge thing that changes our society across so many levels.
I agree. It's the application of the tool. And 20 years ago, who would have thought that we'd be able to do something like this, a study like this that's founded completely in silico. It's also sort of a reflection on the publication process, sort of a side note here. This is a Cell paper, obviously a very high profile paper, but everything that was done here was exclusively in silico or in vitro. And in fact, one of their limitations that they mention is that the study doesn't actually provide any in vivo evidence for the formation of this complex in mammals. Whereas, back in the day, I would feel like for a cell paper, you would have to do that in vivo validation. But I guess it's a reflection on the power of the technology, I suppose.
Again, partner, living in a simulation. What do you need? Validate what? This is it. Oh boy. Listen, talking about simulation, this is a little bit simulated. And it goes to, again, plasticity, as I said at the beginning of the show, and reprogramming. But like kind of a mini reprogramming. And it's coming from Hongqi Deng, who we just covered a story of his group, massive group, but led by him few episodes ago, where they had the chemically induced pluripotent stem cells that they then generated into pancreatic islets that then they transplanted to a patient and pretty much cured them. I mean, that's what he did last month. And this month, kidding, that obviously was the culmination of like literally decades, but big paper just published recently. And this is I think another big paper that is more like conceptual and technical, and a really remarkable indication of things to come. And it's dedifferentiation, but here talking about in like the context of limb generation, like remember the axolotl? Arun, you love the axolotl. You've done, if there's an axolotl story out there, you put it on the show. And I love you for that, because I love it too. But who needs the axolotl anymore, right? In the axolotl, when we used to care about it, these somatic cells, they get spontaneously reprogrammed to this progenitor state that has increased proliferative potential, and then can redifferentiate to make the functional cells, right? Bone, cartilage, and with that, they orchestrate all that process to make a new limb. Pretty amazing. Humans, no, can't do it. There's some regenerative potential, namely the liver, but typically when tissues and organs are injured, you get a scar, right? And there's a lot of reasons for that. I mean, there's, philosophical, I don't know, maybe, but teleological, to like reduce cellular plasticity, right? You restrict the epigenome, maybe because you want to protect the cell identity. You want to avoid cancer, maybe, for example. Whatever the reason, it sucks if you lose a digit or a limb. Worse, I mean, it's a lot to ask to regrow a limb, but I think maybe the principles are there. And this owes to the early observation of the Deng group when they were implementing that chemical reprogramming. There was like this stepwise, first stage they're like this, and then like this, and then like this, and during that process they observed this transient intermediate state at the relatively early stage of reprogramming, where there is increased chromatin accessibility, and activation of early embryonic developmental genes, not pluripotency, but more early embryonic, including enrichment of genes related to limb morphogenesis and development. Okay, so here we go. In this story, they described how they took adult somatic cells, adipose mesenchymal stem cells, or adult skin fibroblasts from human. They did this chemically induced dedifferentiation approach that reverted them to a progenitor-like state that had features of limb bud cells. And then they show they were transcriptomically similar to like the embryonic correlate, established a culture that allowed pretty much indefinite self-renewal and expansion of these cells over long term, and then show that they could re-differentiate them to these osteochondrogenic cells, that are enriched in a lot of the skeletal lineage cell types that derive from those. So this is, I think, another great contribution from the Deng group in a long list. But here, conceptually, I don't know about like regenerating a limb. Yeah, I mean, that maybe seem a little bit far-fetched, but like the orthopedic indications of this, taking a cell instead of having to go like induce pluripotent stem cell, which frankly, talk to any orthopedist, they're going to be like, what are you talking about? Seems cumbersome. The idea of getting this like a little bit halfway reprogrammed and then forward to these skeletal or chondrogenic cell types, like, hey, that seems like a good idea, avoiding the teratoma potential or that, reprogramming other, negative indications from that. So, Arun, I thought this was a really neat story. Tell me your thoughts.
I think, yeah, really interesting story. The clinical part of it, and they allude to this at the very end, I think is a bit far-fetched. Um, because for one, you have to validate that these things are actually doing what they should be doing, and they did demonstrate that, of course, in vivo, and some other studies as well. But that's way more down the road. I think this is much more interesting, just as a developmental angle and a developmental story. Just looking at that concept of regeneration, which of course is heavily impaired in human digits in the human skeletal musculoskeletal system. Maybe there's also an evolutionary element to that. Looking at this in the context of other species as well would be pretty exciting. But yeah, I'm with you. I would probably stray away from the the clinical implications of this because they even mention it here as well. They have to show that these HCI-LBP cells actually it's a mouthful, man. That is a mouthful. They have to show that these things actually closely resemble the real deal, the real early stage embryonic limb bud cells, and they're not identical. And you want to get as close as possible, as with any diff, to the real deal, of course, right?
Yes, I mean, I'm glad you mentioned that because it's really important to underscore that, right? You talk about like the cell intrinsic process is one thing, right? But like a big part of how the axolotl, so I mean, the axolotl, I guess, can stick around for a little while because a big part of how they're able to make that happen, it's the environment that is permissive of that, kind of, I don't call it de novo, but going back to that regenerative point, right? Like if you put the cells on a stump of even of a finger, the tip of a finger, asking those cells to behave and have the coordinated organizational capacity given that like ectopic environments relative to what they're used to as a limb bud in fetal or embryonic development, like that's the other half of this. I would say more than half of it. That's like, as are more complex than the initialism they chose to name these cells, because that, as Arun said, is a mouthful. But I think that, as you said, also the conceptual point here is really strong. And it's sometimes like maybe you can generate cells that don't need to then sort of organize into a digit tip with the bone and all the other stuff, right? Maybe you can de-diff to something that's a little bit more like, modular in and of itself. I don't want to say blood, right? But yeah, blood. Or in like the orthopedic sense, maybe you can like generate bone for grass or something that's more amenable to engineering and incorporating into the existing clinical surgical paradigm. So I shy away from axolotl-like claims, but I think that the clinical potential of this is still very real.
Hey, you mentioned the axolotl. You catch my interest every single time, man. You know how to get me. You know how to get me going. Maybe I'll have to go down the hallway and ask my neighbor here, Dima Shane, what he thinks about this. Of course, he was a guest here on the podcast not too long ago, talking about all things bone regeneration, musculoskeletal regeneration. Check out that episode if you haven't already. Moving on to the last paper of the roundup, and this is again a reflection on the power of reprogramming. We're not talking about chemically induced reprogramming like the Deng lab is focused on here, but we're talking about something that's caught a lot of attention recently, of course, with the work of Juan Carlos Belmonte and Altos Labs. This is this idea of using the Yamanaka factors for potentially rejuvenation. And I don't think Belmonte and the Altos Lab team was involved in this study, as far as I can tell. With the author, actually, just kidding. There is an Altos Labs member here. Not surprising on this particular paper. So of course, Altos Labs has their tentacles everywhere when it comes to using Yamanaka factors for reversing aging. I mean, that's the whole application for them, right? And this is looking at it in the context of the brain, which is a big thing to do. It's I don't know how to describe it. It's conceptually, very high powered. A lot to think about here. This is a cell stem cell paper titled "Expansion of the Neocortex and Protection from Neurodegeneration by In Vivo Transient Reprogramming." So that there it is, right there in the title of the paper. They're using the Yamanaka factors to guard the brain against degeneration over the course of aging, and they also incorporated some disease models here too. So just again, a reflection of the power of the Yamanaka factors. People have demonstrated this, introducing OKSM or whatever related Yamanaka factors into different tissue types. Remember, David Sinclair did something in the eye recently, and there's been other papers that we've covered too. But yeah, that's what it's all about. We know all about the Yamanaka factors here on the Stem Cell Podcast, obviously. I mean, it's the reason why I got into stem cell biology, in addition to the zebrafish, was back when I was at Duke, I heard about iPSCs, which of course are induced by the Yamanaka factors. And that's a big reason why I became a stem cell biologist. And back then, I would have never thought about putting those factors actually in vivo and seeing their impact on restoring or reversing aging potentially. But here's what they're doing: they're exploring the effects of the Yamanaka factors on the brain, and not a whole lot of people have done this. So they induce the Yamanaka factors in the mouse brain in a controlled spatiotemporal manner, in actually two different scenarios: in brain development and also in adult stages in the context of neurodegeneration. So when they did an embryonic induction of the Yamanaka factors, it perturbed the cell identity of both progenitors, neuroprogenitor cells, and neurons, as you might expect. If you're going to throw such a powerful cocktail that early during development, things are going to go probably a little bit haywire, I would say. But they showed that transient and low level expression of Yamanaka factors is tolerated by the cells. So it all kind of depends on the dosing of these really powerful transcription factors, as we're aware of course, right? In these conditions, at the embryonic level, the Yamanaka factor induction led to progenitor expansion, an increased number of upper cortical neurons and glia. And then, and this is the interesting part of it, is they're claiming that this led to enhanced motor and social behavior in adult mice. Now, I don't know if this means that the mice are going to go up and turn on the TV and watch some football or whatnot, but I don't know what sort of claims they want to make. But they're saying that they are perhaps enhanced in terms of their motor and social behavior, at least at the rodent level, I guess. And then they did it on the opposite side, the other side of the coin, right? So they controlled Yamanaka factor induction in adult neurons and adult brains, and it showed that it's tolerated by principal neurons in the adult dorsal hippocampus, and actually prevented the development of several hallmarks of Alzheimer's. Wow. So just by the induction of the Yamanaka factors in this mouse model, they're able to perhaps, shield against this devastating neurodevelopment, neurological disorder that we're all aware of in humans, which is Alzheimer's disease. So again, reversing cognitive decline, reversing altered molecular signatures in a mouse model of Alzheimer's. So this paper is making a lot of big claims. I gotta tell you, we're talking about improving social behavior in mice, maybe making them, quote, smarter by introducing the Yamanaka factors early during development. And then perhaps on the other side of the coin, restoring or alleviating some Alzheimer's disease phenotypes. I don't know, Dalen. It's some big claims being made here in this paper. What do you think?
Yo, B. I mean, wow. Sounds like like a origin story for a Marvel comic book, but I don't know if it's for like the good guy or the bad guy. That's what I'm wondering about. Is this gonna breed evil intent or a superhero? I guess more social. I could use some of that, but like not kind of kidding. We were kind of kidding, I guess, but also it's kind of true as I've thought about it. When we covered that story from Belmonte about the progeria with the OSKM. And I was like, it was like, okay, there's a good litmus test. What's it going to take for me to dose on the Yamanakas? Like progeria. That's a good threshold. If I had progeria, or if someone I love had progeria, I'd be like, all right, give it
Yeah.
to me, or some other similarly like tragic diagnosis in early childhood. And me, like first of all, I'll just say I'm so curious to see the result of this experiment in like primates. I don't know. I have to think about whether it like meets the bar ethics wise and like cruelty wise or whatever. There's a lot of things to consider. I think when you're going into any animal model, frankly, with with the idea of enhancing. But again, like all seriousness, if I'm circling the drain with like dementia, sure, hit me. I would love to see if that made a difference. And like, what's worst case, right? Teratoma in the brain, like, yeah, sure. I'm probably going to die a little bit quicker, maybe not. But like, just I would be curious about whether there's like end stage or compassionate use type indications where this would be tolerated, because I'd be really interested to see on the other side of it. What I'd worry about, we talked about on the show already. That's the theme: plasticity. I think the idea that this is like rejuvenative, I think can be true, but also I think it can create this kind of like, I don't know, a meta state within wherever it hits, where cells are more impressionable and can go off target. And in the brain, you know, maybe you get more social, or maybe you get more something bad. So I think we got to be wary about how we move forward with this, because yeah, like it's easy for me to be like, yeah, if I'm circling the drain, give me the Yamanaka. But like, I wouldn't want that to happen, and it would be really, really awful if then those people, or if someone theoretically who received that had some really, really complex, like neurological, like truly changed their personhood reaction. Like that's a crazy, crazy intervention if that's what it's doing.
Yeah, it's papers like these that make me excited to be in this field because this is borderline science fiction stuff, right? I mean, I thought just the development of iPSCs was science fiction back in the day. I grew up a sci-fi fan, and then all of a sudden you can make pluripotent stem cells from somebody's skin or blood. And here we're talking about using those same factors to make those iPSCs and perhaps turn back the clock on aging, or maybe even turn back the clock on something like Alzheimer's disease, like what you're talking about. The claims are bold, but I agree with you. Just from a curiosity perspective, this is so exciting, and inevitably they're going to be doing the primate studies, and then perhaps those claims of social improvement or motor function can be better validated. Because for me personally, and I don't do a lot of mouse work, making these big claims, these neuro claims about improved mouse behavior is a little wishy-washy to me because I don't know what normal behavior of mice is, and I'm sure a lot of people do. Um, but I would feel, I guess, perhaps a bit more comfortable about the conclusions if they were making some of those conclusions in a primate model, and inevitably, I'm sure that's where they're going to go.
Yeah, that's inevitable. And as you said, what an exciting time to be in the field. And I think I figured out why it's so exciting, Arun. It's because it's a freaking simulation, partner, and that's the way they wrote it. All right, because who would pay attention otherwise? We're about to invite Mary into the chat to talk about this simulated science experience that we're all living through. But before we get there, a quick message from STEMCELL Technologies. Are you looking to advance your liver research with organoids? The Hepatic Organoid Kit can help you easily and efficiently establish, maintain, expand, and differentiate human liver organoids for your experiments. Organoids grown using the Hepatic Organoid Kit are suitable for studying hepatic development, metabolism, regeneration, and disease, and are suitable for studying drug metabolism and drug-associated hepatotoxicity too. Visit us online to learn more about hepaticult at stemcell.com/hepaticult. All right, everybody. Today on the show, we have with us Dr. Meritxell Huch, also known as Mary, who's a scientific director at the Max Planck Institute of Molecular Cell Biology and Genetics, also honorary professor at the Technical University of Dresden. The Huch Lab exploits both the liver as a model of extensive regenerative capacity and the pancreas, which exhibits very little regeneration potential, to unveil the biological processes that control adult tissue homeostasis and repair and their deregulation in disease. The experimental strategy in the lab involves the combined use of animal models and in vitro mouse and human organoid models that recapitulate key aspects of mouse and human liver development, proliferation, and regeneration in culture in a controlled environment. Mary, thank you so much for joining us on the show today.
Thanks for inviting me to be here today.
It really is a pleasure to have you. Again, welcome. As I mentioned there in the open, you focus on liver, pancreas development, and regeneration, but I feel like these organs are more platforms for you and your lab for asking fundamental questions about intercellular and niche cell communication in the context of development, regeneration, and disease. Why don't you give us an idea of what your interests are in your own words?
So I've always been fascinated by the ability of tissues to repair after damage, especially the liver. I've always been fascinated with the idea that you can cut part of the liver and it will regrow. And I was even more fascinated with when I realized that we know not much about it. We know some about it, but we don't know some processes, how the liver senses the damage and replies to it. And we even know less in human individuals, right? Because we cannot start chopping the livers of people just for the sake of understanding how that works. And despite this has been a question that even the ancient Greeks have been interested in, and we've learned a lot from animal models, there's still questions that we still don't know what is universal, that we've learned from the animal models in humans, and what is human specific. We know now that our metabolism is different than the mouse, where we have most of the information. We also know that we even get virus infection is different in the liver than in the mouse. So one of the questions we really like to address is how the human tissues repair, and particularly we use the liver as a model to address that question. And as I said, because we cannot start chopping the organs of people, this is where we use the organoids, as you very well said, Dylan, to ask those questions, particularly in the human setting. Why do we use the pancreas for is we always try to have this comparison from an organ that regenerates well versus an organ that actually has developmentally the same origin from very similar progenitors and the developing embryo, but actually completely contrary to the liver, it does not repair. So we could develop organoid models for both, for liver and pancreas, back in the days when I was in Hans Clever's lab. And we are using them as a tool to address questions along those lines. We've also developed disease models, in particularly cancer models for liver. And we are also using and trying to make the models more complex so we can not only have these epithelial models and ask only questions specific to the epithelia, but also ask questions more on a holistic manner, trying to understand the communication between the cells, why some cells might be important or not for the repair of the tissue. And that's why we also, part of the lab, works on making them more complex.
Yeah, I mean, we're, I think, truly in the golden age of organoids, to which, of course, you and your lab are undoubtedly contributing. And part of what makes this a golden age in organoids is how accessible they've become thanks to standardized protocols that allow labs from really around the world to adapt these cultures and adapt, primary and iPSC-derived organoids across multiple tissue types for everyday use in their lab. Like, my lab focuses on cardiac tissue. There's examples of this across the world, right? And on the topic of protocols, you actually put together an article for STAR Protocols recently on the generation of these liver mesenchyme and ductal cell organoid co-cultures. So maybe tell us a little bit more about that, and also just more broadly the importance of standardizing organoid protocols to really just make them more accessible.
So what we observed back in the days when my lab was not here in the Max Planck, but when my lab was in Cambridge, when I was a junior group leader, we wanted already to start asking the question, what is the interaction between the niche and the epithelia, the mesenchymal niche and the epithelia, to be precise, in the portal region of the liver, which is a particular region on the organ. And we tried many protocols where we were trying to put the cells together. But every time what we were observing is that the mesenchymal cells, they love the plastic, they love attached to the plate, while the organoids, of course, they are on the beautiful scaffold of matrix, and they were totally separated apart, despite you would have them on the same well. The cells, one population, the mesenchyme will be on the bottom, the organoids would be on the matrix. But that was not recapitulating the in vivo architecture, where the mesenchyme and the epithelia in vivo, they are in close contact to the point that they establish cell-cell contacts. So actually, fantastic, talented PhD student in the lab, Lucia Cordero Espinosa, she teamed up with the lab of Florian Hohlfelder, actually literally next door in the biochemistry department in Cambridge, this lab with Timo Koller, they were experts, and they are experts on microfluidics. And it all boiled down to be able to have the cells in close proximity, so they would find each other and interact. And the moment they found each other, they indeed established these cell contacts that resemble the contacts in the tissue. This is what this STAR Protocols paper is about. So it shows a protocol where we standardized how to mix mesenchyme and epithelia from the liver, from the portal region of the liver together. And the trick is just to put them in close proximity. And the microfluidics is just helping us to make microgels, microwells, or microbubbles, let's say, in that case, we were embedding them in a bubble of agarose, where the two cells will be confined in a restricted space of 70 microns. So we were forcing them to be together. And once they established contact, then they could start generating these more complex co-cultures of epithelia mesenchyme, where the mesenchyme is touching the epithelia from the basal side of the cell, which is exactly how it is the orientation in the tissue. Of course, following from here, we are trying to increase the complexity of these cultures, and we are starting to move a little bit away from microfluidics and trying to use also aspects of the self-organization of the tissue itself. We have to remember that, and that's why we study regeneration. In regeneration, the tissue knows how to establish these contacts back. They are lost, and the tissue is able to regain them in vivo. So this is what we try to mimic in vitro. So try to allow the cells to self-organize in a way that they recover the tissue in a dish. But of course, close proximity seems to be very important.
Yeah, as Arun said, this is the golden age of organoids, and I think a large part of it is defined by this, what you're describing, whereas the kind of earlier ages have been about recipes and trying to understand really what the purview and the limits of differentiation, function, all that. Now we're leveraging these kind of engineering approaches, right, to establish the architecture and the complexity, and a large part of that is marrying these engineering approaches with like this let it grow approach, the kind of stem cell intrinsic self-organization capacity, and it's so fascinating to see how those two really kind of dissonant, I would say, forces are married. And I mean, taking it back, I mean, we're here at the point of the spear talking to you But taking it back to another era of your own creative process, I want to take it back to this story you had in Cell, Mary, when you were still in Cleaver's lab. And the story is about isolating these bipotent stem cells from adult liver. And I've always thought that the liver of all organs would be this platform, really amenable to a therapeutic approach that leveraged that regenerative capacity in a kind of sort of biopsy expansion and then auto transplantation paradigm. But here we are. I mean, I don't know that really has, well, you can tell me, where are we with this idea of like an adult stem cell, really something I would say that is the most akin to blood, which has been a resounding success therapeutically as an adult stem cell. Like where is the liver there? What are the challenges? And do you think that is going to be the way we realize this sort of, exogenous cell expansion therapeutic liver approach?
Yeah, this is a very interesting question. Of course, at the very beginning, we're talking about the Cell paper you're talking about is from 2015. At the very beginning, we were already happy that something was growing. Some human cells were growing. I mean, we have to remember that we had the Hayflick limit as the dogma saying that human cells, non-transformed, cannot expand beyond 5 passages unless they get fibroblasts, unless you are a fibroblast. So basically, epithelial cells were not able to expand. And we were already very happy to see that we could expand epithelial liver cells beyond this Hayflick limit for months in culture, retaining the genetic stability. And of course, then the immediate question was exactly what you pointed out, right? Can we use them for cell therapy transplantation? We know that many liver diseases, especially monogenic liver diseases, they would benefit. So they require liver transplant in order to cure the patient. And they would benefit tremendously if we could perform what we do in mice, which is if we transplant hepatocytes into mice, we can rescue many phenotypes. Particularly, for instance, a classical example is the mouse model from Marcus Grompe's lab, developed by Marcus Grompe's lab, and this tyrosinemia type 1 mouse liver disease model. You transplant certain number of hepatocytes, fresh isolated hepatocytes, the liver recovers, and you recover the function, you recover the mice from a lethal disease. That would be the $1 million question that you're asking, how we could do the same with grown cells expanded ex vivo from a human, expanded in the petri dish, in the culture, and put them back into another human and cure this by cell therapy transplantation, or at least extend the life until they can get a liver transplant, right? How far are we there? I think in the Cell paper, we gave a proof of concept that this is possible, but extremely inefficient, to the point that it would not be feasible in humans at the moment. What is the reason for that? I would argue the cells that we transplant, they are not hepatocytes. They are not because hepatocytes can do it. So it means that there is the potential, but we are not there yet. And only when we will get cells of a mature state, that is mature enough to resemble mature hepatocyte, then maybe we are going to be able to start thinking on cell therapy transplantation or cell therapy restoring cells in human livers. Of course, before humans, we have to do all the preclinical studies, and we are still not there yet. This is what I'm trying to say. Having said that, it's true that there is a lot of work done around the world trying actually to improve these systems and also growing hepatocytes directly. So Hans Clevers himself showed that he was able to, and Hui Hu in his lab, they were able to expand fetal liver as a hepatoblast-like cell that they could transplant into a mouse. And again, they saw the engraftment, which was much better than that was in 2018. And it was already an improvement from my earlier protocol from 2015. So we're slightly moving there. I think the future is bright and we are gonna be getting there, maybe sooner than what we think. But at present, we are not there yet.
Yeah, we're getting closer to the goal, as you're alluding to, and it seems like we're running into this almost universal problem in the stem cell field of cell maturity, right? I mean, the cardiac cell transplantation field has the same issue. How do you make sure the cells you generate and ultimately transplant will integrate with the host myocardium? I think every stem cell therapy that's in the works has to deal with this. You alluded to Hans Cleaver's, the lab that you trained in, and the incredible scientist himself, who has, of course, been this amazing leader and pioneer in the organoid field. He certainly continues to publish amazing stories in this area, and it seems like we cover a new paper from his lab almost every week here on the podcast. That's what it seems like. So tell us more about that formative time in your career training in the Cleaver's lab. What was it like to be in that environment?
Oh, that's so when I joined Hans' lab I interviewed in December 2007. So they had just published the LGR5 small intestinal small intestine stem cell paper, where LGR5 is shown to be stem cell in the gut by Nick Barker. And I was fascinated by the power of being able to trace and see these stem cells that you could prove for the first time the stem cell in the gut. I was very, very, very fascinated by that. And then when I joined the lab, I joined actually in September 2008. And of course, the lab had already been moving. And Toshiro Sato, a former colleague of mine, he had generated the small intestinal organoids, and he was testing them and trying to see whether he could even start from one cell. And I was fascinated by that, to the point that I thought, okay, can we establish other models? Can we do other tissues? And I went on, and then I did the stomach, and then as the first one, and then the liver and the pancreas. And I have to say that the time there was extreme time of it seemed that the constant discovery, it was constantly doing new things that nobody had seen before. And it was a real joy, a complete joy. I enjoyed every minute I was there. Of course, was a lot of hard work and sometimes a lot of disappointment, especially at the beginning when things were not growing. And because I started with the naive idea that we use the same small intestine media, kind of a universal stem cell media. We didn't want to do organoids, and we ended up with stem cells making morphogenesis, these beautiful buddings of the small intestine, the same for the glandular domains of the stomach. So I was really fascinated. I am, when I look at them, these beautiful buddings of the small intestine or the stomach organoids. And then I had actually trained myself doing liver cancer and liver metastasis and pancreas cancer. So I very immediately had this wish to move to the organs that I was trained with, which was liver and pancreas. I remember actually saying to Hans, I want to do pancreas organoids. And then he said, we are not a pancreas lab. You better start with something we know. And that's how I started with the stomach. And then eventually we did the other two, the liver and the pancreas, as well. Very intense, I would say. It's a very intense lab, but it was very intense in terms of the science. It was a fantastic brainstorming every day between the people in the lab. It was I actually have a fantastic memory from my training time as a postdoc, to be fully honest. I really enjoyed, I think, every minute of it.
That's such a great look, a great peek into that time, which really was a transformative time for science, and you were at ground zero. I mean, you could argue that that's the most influential idea of the 21st century so far, is the organoid and stem cell differentiation. And I think it's great to hear from your perspective as someone who really was the part of building that out. There was that idea, and you needed such a force of people to say, what about this? What about this? What about this? And to look at it in all these different contexts and build out this concept of organoids across all these domains. and you representative amongst many others of that like organoid diaspora, the diasporoid from the Cleavers lab that really has resonated, in such a tremendous way. So thanks for giving us a peek there. And you mentioned there how you wanted to leverage that. You took your ideas and your training and your interests to the organoid platform. And I want to dig in there a little bit about your application of liver organoids to elucidating pathological mechanisms. In this case here, I'm referring to your recent Nature Communications paper that knocked out specific factors to integrate their role in the emergence and progression of liver cancer. Can you elaborate on that story for us a bit, and how those insights can then be leveraged to address human disease?
Yeah, so that story started as I would say, as a collaborative effort between myself, Bong Kyung-koo, who is now in Korea, but he was back that time. He was in the Stem Cell Institute in Cambridge, and next door where I was at the Gurdon Institute. He was at the Stem Cell Institute, and we actually had both trained with Hans, and we know each other from our postdoc, and he had. Had a lot of interest on Wnt pathway, on these negative regulators of the Wnt pathway. And I'm having my interest on liver and liver regeneration. And it turned out that while we having a discussion, then he said, these two genes are mutated in liver cancer. Do you want to look at them? Maybe it's something important in the liver. Nobody had looked at what is their function in the liver. I said, okay, just he had the mice, I had the Cree driver to knock out these genes on the liver. We just started one cross and we had one shared PhD student, and let's see how it goes. And actually the phenotype was very surprising. We were expecting the mice to not be able to regenerate, but we didn't expect much. And we expected that they would make a cancer very soon. But actually, we very soon realized that the mice had a metabolic phenotype, which was not expected. So that story at the end, we had to go in depth and try to understand it. And it's a mouse model. When you mutate these two genes in the mouse liver specifically, eventually they develop cancer, but this is not a driver for the cancer. What we found out is that the liver becomes steatotic, it becomes fatty. And fatty liver is a predisposing factor, not necessarily will induce cancer, but is a predisposing factor for having liver cancer. So, and when we induce damage to these mice, then they indeed develop liver cancer. So we went on into the mechanism, and when we were puzzled is that we could not detangle what happens first is that the change on we are mutating only the epithelia, but we see a dramatic phenotype in the tissue where there is a lot of stroma reaction, there is fibrosis, there is immune infiltration. And in order to detangle what is first, or whether there is a direct impact on the epithelia, or is actually because you are having epithelia that is unhappy, then you're having a reaction from the niche, we took advantage of the organoids. And then we generated organoids from these mice to detangle the question, is there any impact of mutating these genes in a cell autonomous manner, or is actually not? Is it actually the fact that the cells, because what happens when you mutate this gene, you get Wnt activation. We knew that Wnt is a driver of proliferation in hepatocytes. So is it because the hepatocytes proliferate and then that changes the niche, or is something that is intrinsically happening to the cells, to the hepatocytes? And actually, the organoids allowed us to answer part of the question. We still could be a combination. But at least for sure, we know that mutation on the epithelia, if you mutate these two genes in the epithelia, you get an upregulation of a lot of lipid genes involved in lipid metabolism at the transcriptional level. And what it eventually happens is that the hepatocytes start accumulating lipids and lipid droplets. And that eventually, we think, we didn't prove that that's exactly what happens, but it's known by other mechanisms that accumulation of lipid droplets in hepatocytes causes lipotoxicity and hepatocyte death. And then that results in vivo, and what we think is in a feedback loop whereby the accumulation of lipids in hepatocytes and the steatosis in the hepatocytes is triggering hepatocyte death, and then this constant damage regeneration response of the liver trying to recover from this damage. And eventually that predisposes to the cancer. So the organoids played a crucial role to understand and detangle cell autonomous versus non-cell autonomous mechanisms.
Yeah, I think that's really a strength of these organoids. I mean, they're great model systems for studying disease mechanisms, but both cell intrinsic and extrinsic mechanisms, like what you're alluding to. On the IPS side of things, they're very powerful for recapitulating developmental mechanisms in a dish as well. So it's almost like two sides of the same coin there. On the topic of organoids, it seems like we're just making more and more complex organoids these days with incorporating things like vasculature, even combining different organoid types together to create these multi-tissue assembloid approaches. For example, we've often covered Sergio Pasca's work on the show on brain assembloids. When it comes to making liver organoids and organoids in general more complex and overall just improving the models, what are the first things that come to mind for you? Like, what will take these amazing organoid models, as they already are, just to the next level for in vitro modeling?
I think one aspect that is very important is recapitulating architecture. And part of my lab is working on that front, but I think it would apply as well to the brain, because it's also the architecture is very specific. So the beauty of the gut or the intestinal organoids is that it's a single layer epithelium. In a sense, it's the simplest that you can get. Single layer epithelium, and then you have the stroma of course, on the basal side, but architecturally is quite simple. If you compare that to the architecture of the liver, for instance, in the liver you have mixed up epithelia and stroma in a particular way. So the hepatocytes and I always explain that like the liver is an organ that is a two-pipe system where you have one pipe that is having the blood and the other pipe that is driving the bile, and they cannot intermingle. So you have these networks of sinusoidal or vasculature and the network of the bile canaliculi where the bile is released, and they cannot intermingle. And that's terrible. That's really causing disease. And this is basically an engineering problem, and which the intestine in that sense doesn't have. So recapitulate, and this architecture is critical for the function. So hepatocytes have to release the bile into the bile canaliculi. The bile canaliculi is a 1 micron diameter, not even, it's a tube, it's lined by the apical domain of the hepatocytes in 3D. Then it releases this bile into the bile duct, which is lined by bile duct cells, by cholangiocytes. And the blood goes from the vessels all the way from the portal to the central vein, the hepatic vein. And one hepatocyte will be facing in one side the endothelium, and on the other side is facing the bile canaliculi, right? But it's opening the lumen, which is facing the other hepatocyte. This masterpiece of engineering is a difficult thing. This is, I think, the grand challenge, to recapitulate this architecture. Of course, to recover function, it might not be needed. Basically, maybe we can recover function of a diseased liver. From all these monogenic diseases we discussed before, you just by recovering 30% of the function, you already can rescue the phenotype. But if we want to engineer liver in a dish, our grand challenge is the architecture, is recapitulating this architecture so you still have endothelium in one side, bile canaliculi and another hepatocyte on the other side. So hepatocytes are sandwiched between the endothelium, and they have to connect the bile duct. We're starting to get there a little bit, not with endothelium, maybe a little bit with, there is, so the group of Tanemitsu and Taniguchi in Japan, they had a paper in Nature Comes where they combined cholangiocytes with 2D hepatocytes, and they started to see a little bit of the connection between this monolayer of hepatocytes and some cholangiocytes that they have in the dish. But of course, they don't, being a monolayer of hepatocytes, you don't have the three-dimensional architecture of the bile canaliculi. So, which is a three-dimensional network, right? So there is starting to be things that are starting to look into that direction. We're also working strongly on this front, and maybe we can soon share things on that direction. Because one of the things that we have observed is that in organoids, because they are three-dimensional, now you can dream on the idea of having three-dimensional networks of bulk and nuclei. We showed that recently, still at the ISSCR, when there was the meeting, I presented some unpublished data from the lab along those lines. And of course, then that opens a possibility to start dreaming about making it more complex.
Yeah, again, reminding me of this in this new era of stem cell science, biology. It's really about that meta function, right? It's not the cellular function, but the architectural assembly and coordination of function. It's not enough to have the cells and have them be there. They have to work together, right? It's a good metaphor for the effort it's going to take to realize all the therapeutic potential here. It's not enough to have all the specialized knowledge, but all these brilliant scientists have to work together. And that's what you've got there at the Max Planck. This is not the first group leader that we've had on the show from the Max Planck. Most recently, Ludovic Vallier, who's a fellow pioneer in the field of liver development and regeneration. He gave us a nice insight into one of the strengths of the Max Planck, which is the investment in scientists with a long leash for them to pursue the most innovative ideas unhindered by, I guess, kind of the extrinsic, pressures for funding pursuit or the externalities, political, whatever they may be. Tell us about your experience there at the Max Planck. What sets it apart in your view?
Actually, I can only say wonders of the Max Planck and the Max Planck Society, and probably I agree fully with Ludovic that the perspective of coming to an institute where you can have that is, so your position is secure because we have as a scientific director you have permanent position. It allows you to think on risky projects, I like to call it. Risky projects in the sense that, of course, projects that are always, so when I was in a welcome fellow at the Gurdon Institute in Cambridge, I was on a fellowship, meaning that it's a kind of a time limited, and you have to be able to produce, and having projects that go beyond your fellowship is something that is difficult to sustain. But the long-term perspective here gives you the possibility to think on projects that are risky, that maybe even they would be hardly funded by third-party funding, by grants. But if they do work, then this is a great rewarding. A little bit like the ERC funding in Europe, which of course is also finite, right? Has a timeline, but it's a 4 to 5 year funding, like long-term to do risky projects. And I think that's in part what I have found more rewarding from being at the Max Planck. And I must say, I mean, my colleagues here at the Max Planck Institute here in Dresden, at the Max Planck Institute CBG, they are amazing. I mean, my colleagues, I have cell biologists like Marino Zerial, from which I've learned a lot about the cellular polarity and the polarity of the hepatocytes, which has been very important for our work. I have Tony Hyman, works on condensates, and it's always posing questions more at the molecular level, and it's always challenging the molecular understanding of our work. And also my colleague, Anne Pemberton, she's also a pancreas organoid and pioneer, and thinking from the developmental perspective. So we are very complementary, and there is a very collaborative environment. And actually, I am very, very happy of my colleagues and the interactions. And we also now have a new director, which is a mathematician, and we are also starting to think on more like mathematical models or explanations for the different observations that we have.
Yeah, it seems like just a tremendous place to be a scientist, collaborative environment, innovative, all the superlatives that you can think of. And hopefully not the last person we're going to have from the Max Planck on the show, but we've had, a number of folks from the show who have spoken very highly of the institution. I mean, you've alluded to this. You've traveled Europe throughout your training. It doesn't matter where you've been in many places, all in search of great science, right? You started off in Spain, in Barcelona, of course, for your bachelor's, master's, PhD, before going further north to the Netherlands and the Cleavers Lab, and then further north than that, I guess west, starting your lab in Cambridge in the UK before ultimately moving to Germany to the Max Planck. And talk about a Euro trip, right? I mean, it seems like you've been very open to moving around. You've moved around across Europe for the best scientific opportunities, very willing to change countries as well. I mean, this is perhaps a foreign concept for many folks like us here in the United States who are kind of stuck in this big old country of ours, right? So give our listeners, especially those just starting their training, some advice about your approach to, I guess, traveling in pursuit of the best scientific opportunities. Do you have a strategy for that? What was that like?
No, I never had any strategy. And I would say I go where my heart brings me. Probably that's actually the title of a book from my Italian writers, Susanna Tamara. But the truth is that I was lucky enough, or I would say I've been very fortunate to have parents that actually told me to pursue my dreams, and I did. My parents didn't get any. They don't have any university degree, so they really supported every single step when I was a student, when I was young, really, and it was always like, whatever you do, just put your heart on it, and it will always work. That was actually probably one of the best pieces of advice. And to be fully honest, this is what I've done. I met Hans in a conference in Barcelona. I was doing gene therapy before, and had been trying to do stem cells. It applied to many stem cell labs in the UK, especially, and I got even non-replies. It happens a lot when you're looking for a postdoc, especially if you're changing fields. And then I met Hans in a conference, and I was fascinated by what he was presenting, the LGR5 story from Nick Barker. And I just approached him, and he gave me the opportunity. And I remember when I came back from the interview, I said to my partner, now my husband, back then my partner, I said, well, I think if he says that I can go, I have to go, because it would be such a lost opportunity. And I've also been very lucky that, yes, my partner and my husband now has supported me, I would say, to the 150 to 100% on doing everything that I've ever wanted to do. And I think it's very important to have this personal support. I started with my personal support from my parents, but also my partner in life. And otherwise, it's very difficult. You have to make if you have to make personal versus. So you have to choose between your passions, right? You have to choose between your passion in science and your passion for your personal life. That's tough. That's probably tough. I was very lucky. I didn't have ever to choose. I could always do what I wanted. And I've been always had the support of my family and my partner with me and my husband with me. Yeah. So I would say that in a sense, I went all over Europe just because it's where I found this is what I wanted to do. This was the fun. I actually only interviewed to one place for a postdoc. And I went there, and I interviewed 2 places for the group leader, and I went to one of the 2. And I was recruited here at the Max Planck by the directorium here at the Max Planck Institute. And it was, again, always a decision of like, okay, I think it would be great if we could go there, and always getting a yes as an answer. So I think it's very important not to have to choose between the personal life and the passion that you have. Yes, I've been lucky there.
Absolutely. And I would say that goes for all of us.
Absolutely.
Not just even in science. I mean, whether your career is taking a Euro trip or if you've spent the last almost 20 years in one institute like myself, you need the support of those around you, you know, in your life, also professionally, to help carry you through the vicissitudes, right? It's not just moving around, but just the swings of science. It really can be tough as much as they're exhilarating. And I thank you there for slipping in. We have some science peripheral stuff we like to talk about after, but you already answered one of those questions, which was a great piece of advice, which I think is great. It sounds like straightforward and simple, but it is so central, right? It is put your heart in it, with another way of saying do what you love. And you know that the idea there is deeper than you think, right? Because you gotta love it, and right. And people say, how do I find what I love? And it'd be sooner ask the question, how do you find the person you love? You go looking, right? And if you find something you love, you know it, and that love can carry you through. So your heart being in the work, that's a great piece of advice. To close us out, why don't you give us an answer to one last science peripheral question? In an alternate universe where Mary's not a practicing scientist with all the innovations, what do you think you would be doing with your life?
Yes, I would probably, I would still try to see where my heart is. I really love dancing. Very few people knows that. And I would really, if I would have had another life, I would have loved to train as a ballerina professionally. And I just find so amazing what they can do with their bodies. It's just pushing the body to extreme that goes beyond science understanding, in a sense. I was actually last week in a conference in Malta, and there was a statue of a ballerina on a particular position, like with the perfect split on the air, and it was and the feet looking like all perfectly aligned. Of course, it's a statue, but of course, it's a statue of someone that at some point can do that. I find that fascinating. I would, if I would not be a scientist, I would really like to be able to dance at that level. Pushing again boundaries, I guess, is what, and because I think my heart will also be there. I'm not saying but this requires a particular skill set of, also your body has to be on a particular, this type of talent. It's something you have to be born with. Yeah.
Yes, and needs to be tuned by discipline, which I think is something that you're quite familiar with. So I think if you can move through space the way you move through the science, that would certainly be a sight to see. I would like to visit that alternate universe. Mary, thanks again so much for sharing all this with our listeners. It's been really delightful.
Pleasure has been mine. Seriously, I've really enjoyed it.
Well, we appreciate you very much. And until the next time we have you on the show, thanks for joining.
Thanks to you for inviting again. Thanks.
All right, you guys, that brings us to the end of this simulation execute terminal program. Just kidding. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL or via email at info@stemcellpodcast.com with feedback or to suggest guests. Until the next time, thank you guys so much for listening.